Mars is an active killing machine. The physics and chemistry of this environment make death the default outcome for any unprotected biological organism. This is the Mars Science Laboratory rover, the instrument that measured a radiation dose of 1.8 millisieverts per day during its transit. On the surface, galactic cosmic rays, high-energy atomic nuclei traveling at near light speed, would cause catastrophic double-strand DNA breaks and organ failure within months for an unprotected human. Even if the radiation is managed, the thermal cycling is brutal. Temperatures that briefly reach 20 degrees Celsius at the equator can drop to -60 in hours. These rapid freeze-thaw cycles create microscopic ice crystals that expand and violently shatter biological membranes, irreversibly destroying cellular architecture. Compounding this is a near-vacuum atmosphere that triggers explosive decompression. Furthermore, the Martian regolith is saturated with perchlorates, toxic salts that destroy thyroid function and kill bacteria on contact. External hardware, like pressurized habitats and spacesuits, are temporary band-aids. Sustaining a long-term presence on Mars relies on a biological solution to overcome these environmental barriers. This microscopic scan shows the 8-legged tardigrade, an organism measuring only a fraction of a millimeter. Despite its size, it has survived all 5 mass extinction events over the last 530 million years. It thrives in conditions that annihilate almost all other life. It survives temperatures near absolute zero, immense gamma-ray doses, and even the hard vacuum and direct UV radiation of open space. When water disappears or radiation intensifies, the tardigrade enters a dormant ton state. It retracts its limbs, expels 97% of its body water, and reduces its metabolism to near zero. This invincibility is a product of active molecular engineering. The tardigrade deploys specific proteins to functionally hit pause on its own biology, achieving a state of suspended animation. The engine of this survival is a class of tardigrade-specific disordered proteins, or TDPs. These molecules lack a fixed shape, allowing them to adapt rapidly as the organism begins to dry out. As intracellular water drops, TDP molecules link into a homogeneous gel. This undergoes a glass transition into biological glass, perfectly trapping cellular components. Reactive oxygen species are completely frozen, preventing DNA damage, while TDP molecules physically wrap and stabilize the DNA double helix. Vitrification stops biological time. By neutralizing the mechanisms of cellular destruction, it allows the tardigrade to remain dormant for decades before reviving upon rehydration. A 2019 study confirmed that this is a transferable technology. When CAS proteins, a specific family of TDPs, were expressed in human cells, they reduced cell death from desiccation by 40%. This provides a pathway for Martian agriculture. Engineering these proteins into staple crops and nitrogen-fixing bacteria would allow them to survive in partially shielded environments with far lower infrastructure requirements. There are hardware applications for this as well. Synthetic bioglass analogs can be integrated into spacesuit liners to scatter secondary radiation, while vitrification protocols can preserve blood and medical supplies for multi-year missions without complex refrigeration. This diagram shows a specific genetic sequence being spliced into a human DNA strand. Using CRISPR, researchers aim to conditionally integrate tardigrade genes into human stem cells, allowing the body to trigger its own vitrification protocols during high-radiation solar events. Deep space survival is moving beyond the limits of thick metal shielding. Success now depends on completely rewriting the cellular resilience of the human body to withstand the Martian environment directly. We are looking at a historical shift where we borrow a 4-billion-year-old biological blueprint to move beyond our home planet. The timeline for this technology is rapidly accelerating. Functional expression of biological glass in human cells is conservatively estimated to be viable within 15 years, well within the projected window for the first crewed landings. The technical barriers to deep space survival are falling. As the possibility of engineering these traits into ourselves becomes a reality, the remaining hurdle is the ethical and regulatory framework for modifying human biology to suit a life among the stars.
Could life survive on Mars? These microscopic water bears just proved it might be possible. When hit with cosmic radiation or freezing temps, their DNA gets shielded instantly. If we can copy this bioglass, humans might just conquer the red planet. Would you go to Mars?